11.1 Experiments of Gregor Mendel. Every living thing has a set of characteristics inherited from its parent or parents. The scientific study of this biological inheritance is genetics. Genetics is the key to understanding what makes each organism unique. The modern science of genetics was started by a monk named Gregor Mendel. Mendel was in charge of garden in the monastery where he lived. In this garden, he did work that changed biology forever. Mendel used ordinary garden peas in his studies. He used peas partly because peas are small and easy to grow. A single pea plant can make hundreds of offspring. Also, pea plants grow quickly and have many traits that are easy to see. The role of fertilization. When Mendel started his experiments, he knew that the male part of each flower makes pollen. Pollen contains the plant's male reproductive cells, called sperm. Mendel also knew that the female part of each flower makes reproductive cells called eggs. During sexual reproduction, male and female made by self-pollination has fertilization to make a new cell. In peas, this new cell develops into a tiny embryo wrapped inside a seed. The male and female reproductive cells of pea plants are in every pea flower. Since pea flowers have both kinds of cells, a pea flower normally pollinates itself. A plant grown from a seed made by self-pollination has just one parent, because both reproductive cells came from the same plant. True breeding plants. Mendel's monastery garden had several stocks of pea plants. These plants were true breeding. True breeding plants are self-pollinating. They make offspring identical to themselves. The traits in each generation are the same. A trait is a specific characteristic, such as seed color or plant height, of an individual. A trait may vary from one individual to another. For example, one stalk of Mendel's seeds made only tall plants. Another stalk made only short ones. One stalk made only green seeds. Another stalk made only yellow seeds. Flowers have male parts and female parts. During fertilization, pollen is transferred from the male parts to the female parts. Crossbreeding. Mendel wanted to learn more about how traits are passed from parent to offspring, so he used pollen from one stalk of plants to fertilize the female parts of flowers from other stalks of plants. This process is called cross-pollination and produces a plant that has different parents. Mendel studied 7 different traits of pea plants. Each of the traits had 2 contrasting forms. Seed color is one example of a trait Mendel studied. The seeds were either green or yellow. Mendel wanted to see what would happen if he made a plant from 2 parents that had different traits. The offspring of crosses between parents with different traits are called hybrids. 2 conclusions. When a yellow seed plant was crossed with a green seed plant, all the offspring produced yellow seeds. It seemed that the green trait had disappeared. The same thing happened with each of the other traits. Mendel drew 2 conclusions from this. Passing traits to offspring. First, Mendel found that an individual's characteristics are controlled by factors that are passed from parent to offspring. Today, scientists call these factors genes. Each of the traits Mendel studied was controlled by a single gene that existed in 2 different forms. Each form of the gene controlled one form of a trait. For example, the gene for plant height came in one form that made tall plants, and another form that made short plants. The different forms of the same gene are called alleles. Dominant and recessive alleles. Mendel's second conclusion explains why some of the traits seem to disappear in the offspring. This conclusion is called the principle of dominance. This principle states that some alleles are dominant recessive. An organism with at least one dominant allele for a form of a trait will show that form of the trait. An organism with a recessive allele for a form of a trait will show that form only when the dominant allele for the trait is not there. In Mendel's experiments, the allele for tall plants was dominant, and the allele for short plants was recessive. Similarly, the allele for yellow seeds was dominant over the recessive allele for green seeds. Key question: where does an organism get its unique characteristics? An individual's unique characteristics are determined by factors that are passed from parent to offspring. Build vocabulary. Genetics, the scientific study of heredity. Fertilization, the process in sexual reproduction in which male and female reproductive cells join to form a new cell. Trait, a specific characteristic of an individual. Gene, a factor that is passed from parent to offspring. Allele, one of a number of different forms of a gene. Principle of dominance, states that some alleles are dominant and others are recessive. Word origins, the word fertilization is from the Latin word fertilis, which means to be fruitful or to make offspring. This table shows 3 of the characteristics Mendel studied in his experiments. Build vocabulary. Segregation, the separation of alleles. Gametes, sex cells. Word origins. Like the word segment, the word segregate comes from the Latin root word segmentum, which means a piece cut off. Recessive traits reappeared in the second generation. Segregation. When doing genetic crosses, we call each original pair of plants the P or parental generation. Their offspring are called the F1 generation. Mendel had a question about his F1 generation of pea plants. Had the recessive alleles simply disappeared, or were they still in the new plants? To find out, he had all 7 kinds of F1 hybrids self-pollinate. The offspring of an F1 cross are called the F2 generation. So Mendel crossed the F1 generation with itself to make the F2 offspring. Recessive traits reappear. Mendel found that the traits controlled by the recessive alleles reappeared in the second generation. About 1/4 of the F2 plants showed the trait controlled by the recessive allele. Why did the recessive alleles seem to disappear in the F1 generation and then reappear in the F2 generation? Explaining the F1 cross. Mendel figured out that only the dominant alleles determine the traits of the F1 generation. However, the trait controlled by the recessive allele did show up in some of the F2 second generation of plants. So Mendel realized that the allele for shortness had separated from the allele for tallness. When did this separation or segregation of alleles happen? Mendel thought that the alleles for tallness and shortness in the F1 plants must have segregated from each other during the formation of the gametes. Gametes are the sperm and egg cells that combine during fertilization. Making gametes. Was Mendel right? Let's assume that the tall F1 plants inherited two alleles controlling their height. They inherited an allele for tallness from their tall parent and an allele for shortness from their short parent. Because the allele for tallness is dominant, all the F1 plants are tall. When each parent makes gametes, the alleles for each gene segregate from one another. Therefore, each gamete carries only one allele for each gene. So each F1 plant makes two kinds of gametes. Some have the tall allele, and some have the short allele. Build vocabulary. Recessive traits appeared in the second generation when the first generation of plants were allowed to self-pollinate. Look at the segregation diagram to see how alleles separate during gamete formation and then pair up again in the F2 generation. A capital letter shows a dominant allele. A lowercase letter shows a recessive allele. Now we can see why the recessive trait for height, little t, reappeared in Mendel's F2 generation. Each F1 plant in Mendel's cross made two kinds of gametes. Some had the allele for tallness, and some had the allele for shortness. Whenever a gamete that carried the little t allele paired with another gamete that carried the little t allele to make an F2 plant, that F2 plant was short. Every time a gamete carrying the big T allele paired with another gamete carrying either the big T or little t allele, they made a tall plant. In other words, the F2 generation had new combinations of alleles. Key question: how are different forms of a gene passed to offspring? When gametes are made, the alleles for each trait separate from each other. That way, each gamete carries only one allele from each gene. When gametes are made, the two alleles of each gene separate so that each gamete carries only a single copy of each gene. Each F1 plant makes two types of gametes, those with an allele for tallness and those with an allele for shortness.